The next talk is by Dr. David Saloner from UCSF, who I was lucky enough to work with during my time there. It's on advances in MRI or carotid MRI, promising techniques for clinical translation.
And I use this to advance.
Yes.
So I want to thank the speakers, the organizers for this opportunity to give this talk on potential clinical benefits that might accrue from MR angiography. And I'm going to focus mostly on the extracranial carotid arteries. I have no financial conflicts to disclose. So I'd like to go back to the start of this. We used to be the MR Angio Club, but now the Society. 34 years ago, when extracranial carotid artery was probably the key organ system that our colleagues were all focused on investigating, and it was those developments were happening simultaneously with the conduct of some of the major trials to compare the efficacy of medical treatment versus surgery in treatment of extracranial carotid artery disease. In fact, if you go back and look at our early logo for the society, it was of the extracranial carotid bifurcation. And so at that time, interventional guidelines were developed, and they've sort of been set in stone, even though that stone has been eroding over the years, particularly because of the advanced use of statins that was not part of the cohort of those original trials, and the changes in lifestyle that patients are encouraged to adopt if they are suspected of having extracranial artery disease. But I think at the time, our mission was to, as accurately as we possibly could, determine diameter stenosis. We thought that if we could not develop an MR technique that would differentiate 69.9% stenosis from 70.1% stenosis, we would be a failure. And we spent a lot of time trying to drive that technology, but I'd argue that things have evolved and that the utility of MR is not so much on that exact pinpoint accuracy, but as adjunctive to a lot of things that are happening in the clinic. Clearly, symptomatology is critical. There are a large group of neurologists who would argue never to intervene on anybody who is asymptomatic, no matter the degree of stenosis. So if we then go to the those who are symptomatic, what does symptomatic means? If you're 50 to 59% stenosis and you're more than 2 weeks out from a TIA or an event, you are now thrown back into the asymptomatic category. If you're 70 to 99%, and more than 12 weeks out, it's almost like a de novo startup. And again, we continue to look at luminal geometry as a key indicator for. So where is MR angiography strong? It's strong in symptom workup. We've seen already from the previous talks today that when the patient comes in with maybe some vague symptoms, it is invaluable to rule out any stenosis of the extracranial carotid arteries. So it doesn't need to be precise imaging, but if you acquire a quick time of flight MRA through the carotids and say there is no disease here, that is hugely helpful in the clinical workup. Often when patients are being considered for coronary artery bypass grafting, our clinicians are interested in how are they going to sustain the procedure? Should we be concerned about risk of stroke because of carotid disease, and therefore a quick study of that kind can be very helpful. And it's very helpful in longitudinal monitoring, something that I really care about a lot. I'll give another talk later today showing the use of longitudinal monitoring. It's non-invasive, it's operator independent, and very helpful. Again, in precise diameter stenosis, maybe moderate in that ability compared to CT angiography. One thing to note is that catheter angiography has virtually disappeared as a diagnostic tool, given the availability of both MRA and CTA. And certainly not for population screening, given its expense and the low prevalence. So this is sort of a typical carotid bifurcation scan, less than of the order of 4 minutes, high contrast to noise, high signal to noise, probably still can be motion sensitive and always will be flow dependent. We do have the ability to use contrast, as was shown earlier. You had a study showing actually in pretty exquisite detail the degree of stenosis, sub 30-second acquisition, and motion and flow insensitive. So what are the opportunities that I can think of for advances of clinical value? And I think extending the region of interest rather than focusing on the extracranial carotid bifurcation, but going from the arch to the circle of Willis, as we've heard mentioned, combining it with functional information, and here I'm talking mainly about 4D flow, but there's other methodologies for dense and other things for looking at vascular compliance. And quantitation, I think, will be a key thing to pursue. So generally, the disease of the carotid bifurcation is from 2 centimeters proximal to the bifurcation to 4 centimeters distal. But we know that there is an atherosclerotic burden that extends from the arch all the way out to the middle cerebral arteries. And having an ability to evaluate that entire territory would be of great value. And it is not only, I think, one of the exciting areas that people are starting to talk about more and developing ideas about more is not simply stroke. I don't mean simply, but not only stroke alone, but also for cognitive decline, dementias. What is a contribution of the overall atherosclerotic burden to cognitive decline? And I think that's going to be a very large area of growth. So we've seen images like this of contrast-enhanced MRAs from the arch, but in order to acquire this in the 30-second window that we have to trade off resolution, and this is a 1 millimeter isotropic resolution. We can use intravascular contrast agents such as ferumoxytol, where we could go to a 3-minute acquisition, for example, as in this this case shown here, and get 0.6 millimeter isotropic resolution with outstanding coverage. If you open up the field of view on this image, you will see that I did not carefully carve away the veins from the rest of the anatomy, and this is what we have to deal with. So on the one side where you've cleaned up the venous contributions, you can see that it's very much a problem. On the contralateral side where I did not do that post-processing. So separating arteries and veins is a challenge for using ferumoxytol, and it is an expensive contrast agent, but I think it has huge promise for a variety of applications in the vascular system. Already, we saw Dr. Baradaran showed a case with intraplaque hemorrhage, MP rage, very easy to detect the light bulb signal of this very high-risk feature. But I think extending the region of interest, as is shown by the group, by this publication from Sakai et al., where they've gone all the way from proximal to the bifurcation up to the bifurcation of the middle cerebral artery, and this curved reformat beautifully showing both the black blood lumen and the wall along that entire territory, I think is very promising. And a similar case here from Jai et al. For a cervical artery dissection, showing both the black blood ability to cover a large territory of interest and the MRA, contrast-enhanced MRA. So very rich combined information to look at that presentation. There are a few groups who are quite interested in carotid webs. Carotid webs are fascinating entities. They're just a very little atherosclerosis present, a shelf that seems to sit in the carotid, off the carotid wall, but many patients will present with recurring symptoms that are thought to be referable to these carotid webs. And so here I think using 4D flow we have found to be very helpful and high resolution. Here's a patient. You can see this shelf-like web at the origin of the internal carotid artery, 3D black blood confirming that focal shelf. And then we did a 4D flow study on this. It appears that my 4D flow video did not upload. So, but I can allow me to describe the features. What we have is we have a strong forward antegrade flow, but where we see these particles, we see a strong rotating vortex just at that plane. This is similar to the flow that you might see in an ulceration. And so this woman was thought to have this carotid web. She went to surgery, and indeed at surgery you can see the resected endarterectomy specimen showing this cusp sitting off the wall. And although it's relatively little atheroma per se, it is something that our clinicians are very aggressive in treating and resecting because they keep on throwing thromboemboli. Vessel wall composition is also clearly really a rich area, a target for looking at. We have 2D, 3D techniques. I think the drive here is to go to quantitative measures to look at all the various components of the wall of the carotid artery. I think some of the most exciting work is from the group here at Cedars-Sinai, Zhaoyang Fan, Yibin Ji, and Debiao Li. And here is some images from their work where we see the conventional T1, T2 weighted, contrast enhanced TSC and MRA, but also Q-match, a single sequence acquisition, which you can extract both black blood and bright blood images, but most importantly, extract out these T1 and T2 maps. And if you take that data and plot the T2 values versus the T1 values, what is very exciting and provocative is to see that each of these components separate out, cluster into different groups. So if we have this ability to image patients, characterize on a pixel by pixel basis what the composition of the atheroma is, might we use this to generate a risk index? Clearly that would require recruiting a large number of patients, developing a large database, and having a look at this in regard to an outcome trial. So those are some of the elements that I think we might rely on to push the field forward. The carotid MRA is currently of great value in workup of patients with clinical questions. I think measures of the wall, in addition just to the lumen, provide rich information on the disease. Extending the coverage will give us a more comprehensive evaluation, not only for stroke risk, but for cognitive decline. And tools such as biomechanics and bioinflammatory markers, which I did not dwell on in this talk, remain promising research topics. And I'm quite certain that quantitative metrics will be key for establishing MR as a primary tool for clinical decision making. So thank you for your attention.